Hello, I'm Dr. Sachin Kulkarni and I'm going to take you through a small presentation, very interesting one. And this has come from a Vietnam university and published by Nguyen Hoa, and this is a publication in 2022. And it is titled The Effectiveness of Sperm Preparation Using Density Mini Gradient and Single Layer Centrifugation for Oligospermic Samples. In this particular study, look at the table there. They had oligospermic samples, and each oligospermic sample underwent a semen analysis, a sperm DNA testing by SCD method before the preparation. Then that sample was aliquoted into 0.5, 0.5 and 0.5 ml. The first 0.5 ml went through a mini density gradient, the next 0.5 ml with a single layer 90% density gradient, and next 0.5 ml to 45% density gradient. Then they looked at the washed semen sample and they looked at various parameters like the sperm motility, sperm concentration, sperm morphology, and what is the change in the DNA fragmentation. Very interesting. In this particular study, how was the mini density gradient done? For the mini density gradient, they used 0.5 ml of 90%, 0.5 ml of 45%, and 0.5 ml of semen was put on the top, and then the whole density gradient process was taken place. So basically, the amount of density gradient the semen has to traverse was decreased by about 50%. And when it came to 90%, they just used 0.5 ml of 90% density gradient and semen topped on that, or 0.5 ml of 45% gradient and semen topped on that, and they underwent a 1500 revolutions per minute centrifugation, and the final sperms were obtained. Look at the results. When you talk of sperm count, to the basic sperm count, whenever you use any sperm washing method, the count is going to come down. So that was observed in all three samples, but the total sperm count was still good when they used a 45% single layer density gradient for oligospermic samples. And what about the motility? Look at the motility. The motility was the best with 45% single density layer rather than the mini gradient and that of the 90% single layer. So 45% single density worked the best with higher number and highly motile sperms obtained. So what about the sperm recovery rate? Look at the table there. With single 45% layer, the sperm recovery rate was 76% compared to 48% of 90% layer and 44% with mini density gradient. What about the progressive motile sperm recovery rate? That was again 78% with single 45% 0.5 ml single layer. So standard density gradient may not obtain an optimum sperm because sperms with poor endurance will not survive such long density gradient layers. Single layer will effectively reduce the infection, epithelial cell, blood cells, at the same time offer you a better progressive motile count. I'm sure this will be helpful to all of you. Thank you.